Okay, well, you are going to first hear from panel one for the first half of this evening. When we're going to really talk about, holding hope, the promise of precision medicine. It's great that we have an all-Missouri team here, two from Washington University and one from Kansas City. What we're going to do is hear each of them give a very brief presentation that's going to really illustrate some of the things I started talking about in my remarks, and then we're going to have the rest of the time spent for you asking questions. I think there's some index cards being handed out. You can write your questions, and if there, we also will take questions from the floor if other questions come up and you don't have a card to write it down. So I am going to just briefly introduce each speaker. You can read a little bit more about them here, and I think you mostly want to hear from them at this point and not me. And so the first one is Sesh Cole, who has been in the pediatric department since I was a medical student, and actually was... I was 3 years old. But 3 years old, that's right. He was an awesome teacher, I can tell you. I vividly remember when he was an attending in pediatrics when I was a medical student. So, Sesh, we're going to have you kick this off.
Thanks very much, Eric. So I'd like to thank the St. Louis Academy of Science and the National Human Genome Research Institute for asking me to participate in this panel discussion, which is about establishing a dialogue with the community about a transformational change in medicine that's occurring and that will affect us all. So I am a baby doctor. Unlike my august and esteemed colleagues on the panel, I just take care of babies, usually sick babies. It turns out that many babies who present with problems also present problems that don't fit into precise categories. And so frequently we're left without a diagnosis for these medically mysterious children. And sometimes these children are able to survive and they go on extensive diagnostic odysseys, which are quite expensive. I want to show you two quick examples tonight that will hopefully illustrate both the power of genetics and genomics and also help illustrate a bit about how far we've come since October 1st, 1990. So the first baby is a baby whom we discovered by chance. So this is a baby who was born after the full 9 months, 283 days in European descent pregnancies. And the baby had no complications, and the mom is completely healthy, and she did everything correctly, as far as anybody could know. The baby looked good in the delivery room, and within the first 24 hours after birth, developed very severe breathing problems. Interestingly, the baby has had 3 older brothers who were completely normal and had done well and were healthy. But in 20 years before this baby was born in 1991, so in 1971, the same couple had a daughter who was born after the full 9 months and who developed a mysterious breathing problem. She never left the hospital and died at approximately 8 weeks of age. So when this baby presented to us, it's a little unusual to have a full-term baby have difficulty with lungs, especially as severe as this. And we were concerned that we were not really understanding what was wrong with the baby. We can keep babies like this sort of ticking along with life support, but we really didn't know what the diagnosis was. So we finally ended up surgically taking a small piece of this baby's lung and looking at it under a microscope. And we observed that the appearance of this baby's lung under the microscope and the appearance of his sister's lung under the microscope were very similar and quite unique. And so that observation suggested that what this baby had might have been inherited from mom and dad and might have been inherited, might be the same inheritance problem that the daughter had some years before. And so unlike what we do today, we said, well, what ways do we have to analyze this? And it turned out there are about 19 or 20,000 genes in the genome. We happen to have, strictly by chance, reagents or ways of testing for 3 of these genes. And as you can see here, we looked for activity of each of these genes. Here is here's the baby in question right here. And here are several other babies with other kinds of lung problems. And we look, this is gene 1, this is gene 2, and this is gene 3. And the blackness of these dots represent the activity of the gene. In other words, was the gene working or not? And as you can see, under the baby in question, all the babies seem to have this gene active. All the babies seem to have this gene active. But our baby lacked activity in this particular gene. Now, I would like to tell you that I knew this all along when we did this, and that this was just a confirmation of a hypothesis that we had generated. But this was, we were lucky in this situation. We tried 3 and found 1. And then we further analyzed the genetic code of this particular gene in the babies and in the parents, and we found that it was a defect in this particular gene that led to the breathing problems in this particular family, and we have subsequently gone on to describe this as the first genetic cause of breathing problems in full-term babies. So the good news about this was 20 or 25 years ago, we were lucky. We looked at 3 genes and found 1. Now fast forward to a medical mystery that happened within the past year or two. So this is an example of more recent strategies where rather than looking at a single 1 or 2 or 3 genes in a medically mysterious baby, we actually look at all 19,000 genes at once, or 20,000 genes at once. And this particular couple had 3 successive pregnancies with the same problem in each of the 3 babies. And the principal problem was a severely undergrown brain. So this is the brain, the MRI appearance of a normal baby. And you can see how they're nicely folded, and it's sort of big, and it's got some pieces back here that all look like they're sort of lined up. And here is an MRI of the baby, one of the babies in question. And as you can see, this brain is very small, and you can see it's not folded properly. The back of the brain looks strange, etc. So this baby's brain was about 1/20th the size of a normal brain. In addition, each of the babies who had this brain abnormality were also very much smaller at term than we would have anticipated from a full-term baby. They also had some heart problems and some lung problems, and all 3 babies passed away. And so to discover what the problem was in these babies, rather than just guessing at 3 genes and hoping for the best, we analyzed all the genetic code in all the genes from both the parents and each child, and then we used a computer to compare the genetic code from each of the babies and with the mom and dad, and we realized that all 3 children carried a mistaken genetic code in a single gene that had not been previously linked to any human disease. So one of the big advantages of doing this unbiased discovery is that one can identify problems in genes that haven't been previously described or discovered. However, we found this, but we also were able to look in a large number of public databases. As Eric described, there are many of these being developed now, and we found a 41-year-old woman with the same mutations as these babies had. So wait a second. This was a fatal problem. There's a 41-year-old woman problem. So how could we harmonize these 2 findings? However, we also found that there was a second genetic code problem which was close to the mutated gene. And it's illustrated on this, and it was really facilitated by Todd Drewley in our department. So here's the father who has a normal gene copy, has a mutation, which we discovered, and also has this dotted line, which means a little upstream from this gene, there was a big piece of DNA deleted. Okay? The mother has a normal gene copy and has the mutation, but she does not have the deletion. All 3 of the children had the mutation, and this deletion, and the 41-year-old mother had the mutation, and no deletion. So in other words, what happened to the babies was they got a mutation from the mom and a mutation from the dad and this deletion upstream, and this caused the fatal array of birth defects. And although we cannot intervene on this therapeutically at the present time, this kind of discovery helps to inform future reproductive decisions for this particular family. because we can now do pre-implantation genetic diagnosis to determine whether or not embryonic events may or may not be affected. So there are promises and challenges. As you'll hear, I think, from both Stephen and Elaine, we are now able to do relatively cheap, rapid, individualized evaluation of each person's genetic code. And this deciphering shortens diagnostic odysseys for children with medical mysteries. This personalized medicine will permit individualization of standard treatments and counseling. There are, however, as is being discussed in the other panel, ethical questions concerning access to and uses of just genetic code. What if an insurance company found that this woman was pregnant again and had this particular genetic risk? Would the insurance company want to insure her or not? And finally, environmental impact, interactions between genes, and differences between children and adults are still not well understood. So as Eric said, we have made an enormous amount of progress in the last 25 years. We still have a ways to go. Thank you very much.
Thank you, Sash. So what you can immediately see is from these initial stories how things have just changed so dramatically, and being able to use genomics to solve these medical mysteries. But in Sash's case there wasn't super urgency of figuring this out. He wanted to understand what was wrong with these children and help give advice for those parents who are thinking about reproductive options. But there are other medical settings where maybe you need to get that information really quickly. And so we're fortunate to have Stephen Kingsmore here, came over from Kansas City, who really is a leader now in the field of thinking about how to do things in a setting where you need information quickly. So that's my teaser so he can tell his story. I'm not a Mac person, so I don't know how to turn this.
Go to the next slide.
Oh, the next slide. Oh, right after the other. Oh, excellent. Oh, there you go.
Thank you very much. So I'm from Kansas City. You can tell my accent's a little bit different. It's the other end of Missouri. I came here by Amtrak last night, and that was a lot of fun. And we look forward to seeing you all in the playoffs, God willing. Yeah, that's worth a clap. Okay. So I'm going to take you to a baby in Kansas City. This is a case that we saw in April of 2013. This is baby 487. And so this baby initially came to light in our hospital because Mum had an abnormal blood screen. Mum was pregnant and she had a routine blood test to see if her baby was normal or not, healthy, and that was abnormal. And so that meant that she got to earn a pediatrician early on in her pregnancy course. And as part of that in our hospital, she had imaging of the baby inside her womb. And that showed a number of congenital anomalies. And one of those is shown over on the left. It's called an omphalocele, and it's where the covering of the stomach contents doesn't form correctly. And so the stomach and the intestines, as you can see here, prolapses out into the amniotic cavity. So her